Are you able to hear me? Is it audible and visible? Not audible. Okay now? Okay now it is? Is it audible and visible now? Okay it is? Okay. And we completed the osmosis and osmotic pressure. Okay. Again, to recollect this osmosis, we go through the text, how far the delivery synchronizing with the text, you have to check it. Okay. With regard to the osmosis, as a cell is having cell wall, plant cell, and membrane surrounded by cell wall, and cell wall is permeable, allowing water and the substances dissolved. So cell wall is not going to be a barrier. And plasma membrane and tonoplast is which are the determinants of our osmosis. And cell sap, it is hypertonic. It contributes to what it is solute potential. Because it is hypertonic, it is entry of ions into the cell sap. It is against concentration gradient through tonoplast. That's why it is hypertonic. Okay, it is. And diffusion of solvent through selectively permeable membrane. So what are the driving force for osmosis? Pressure gradient and concentration gradient are the driving force for what it is osmosis. Okay. Diffusion of solvent through selectively permeable membrane is osmosis. This definition, okay. When two solutions of different concentrations separated by selectively permeable membrane, solvent or water flowing from, that is moving from low concentration solution to high concentration solution till equilibrium is reached. Once equilibrium is reached, water potentials are going to be the same but not a concentration. At equilibrium, there is no net movement. There is a movement, vice versa, but there is no net movement. That is one point. And what is the physical experiment to demonstrate osmosis? It is thistle funnel experiment. And what is the biotic experiment? It is potato osmoscope experiment. Okay, it is potato osmoscope. And in case of potato osmoscope, the potato osmoscope at the base, that means a trough or depression made, and in the trough or the depression, sugar solution placed in petri dish having water, and cork it is removed as it is impermeable for what it is water. So there is the entry of what it is water from the petri dish into what the trough or depression in case of the potato tuber. There is a rise in case of column in potato tuber. That is biotic experiment. Okay. So water always moves from higher water potential to lower water potential. Less negative to more negative. High diffusion pressure to low diffusion pressure. Low diffusion pressure deficit to high diffusion pressure deficit. Low osmotic pressure to high osmotic pressure. High solute potential to low solute potential. In thistle funnel, the sugar solution in thistle funnel, in the beaker it is water. Initial level is noted, it is L1. And the mouth of the thistle funnel tied with what it is egg membrane. How we are going to get? By making a hole, removing yolk and albumin, dissolving the shell in dilute HCl, intact membrane, that is semi-permeable membrane. And due to entry of water from beaker to thistle funnel, what increases? Water concentration increases. Water potential increases. And hydrostatic pressure compensates the loss of energy due to addition of what it is solute. If you exert a pressure at the apex, it stops osmosis. Whatever the pressure exerted to stop the osmosis, what we call osmotic pressure, that is contributed by sugar in solution, that is in thistle funnel. High the concentrated solution, more the pressure it is required. Suppose 10 bars pressure, 20 bars pressure in thistle funnel. If you create 10 bars, osmosis continues to occur. 20 bars, it stops. If you create 30 bars pressure, reverse osmosis occur. Okay, that is thistle funnel experiment. And in the box question, the movement of the water from high concentration to low concentration, solutes from its high concentration to low concentration, because diffusion of any substance is its own concentration gradient, not depend upon the concentration gradient of what it is others, high to what it is low. To that extent, we completed. Okay. Next, imbibition. This imbibition, it is observed in dry seeds, hydrophilic colloids, dry woods, lichens, which are non-living. Dry seed, hydrophilic colloid, dry woods and lichens, which are the lichens initially the dry. And the dry or non-living substances which absorb water, which absorb water, and swells in volume. Dry or non-living substances absorb water and swells in volume. What do you call it is? Imbibition. Okay. And this imbibition is a kind of diffusion. Suppose it is assertion. Imbibition is a kind of diffusion. It is right. How you can say based on concentration gradient. Based on concentration gradient. Diffusion is based on what it is? Concentration gradient. Okay. So imbibition is a kind of diffusion. It is right. Reason based on concentration gradient. Okay. And in this imbibent and water, it is to be imbibed. The two things, imbibent and water to be, it is imbibed. Imbibent, the substance involved in imbibition, what do you call it is? Imbibent. Substance involved in imbibition. What do you call it is? Imbibent. Substance to be absorbed, it is water. Okay, to be imbibed, it is water. So, there must be affinity or attraction between imbibent and water to be absorbed. Affinity or attraction, and also water potential gradient must be there between imbibent and water to be what it is absorbed. Are you getting it? So, between the two, there must be affinity, attraction, and water potential difference. Delta psi W, water potential difference. Because water always moving from higher water potential to lower water potential. So, water to be absorbed. The substance involved in imbibition, it is imbibent. Okay, it is. Okay, noted it is. The content on the board noted. Noted or not? Okay. Okay. So, we try to get this point. Affinity or attraction between imbibent and water to be what it is absorbed. Okay. So, it is a kind of diffusion. Absorption of water by dry or non-living substances and swells in volume is nothing but it is imbibition. Okay. and different organic substances, different organic substances having different imbibing capacities. Different organic substances having different imbibing capacities. If you take phycocolloids, that means the substance is obtained from what it is, some seaweeds, phycocolloids. Proteins, starch, cellulose. It is having highest, least. So increasing order of imbibing capacity. If you take proteins, pulses, sizer, starch, cereals, rice, wheat. Okay, phycocolloids, proteins, starch, cellulose. So sizer having high imbibing capacity than what it is rice, because the proteins having high imbibing capacity than what it is starch. Cellulose least, if you take paper, it is least, different imbibing capacities. So earlier you said there must be affinity or attraction between imbibant and water to be absorbed. Suppose that is affinity or attraction. And next it is a difference of water potential gradient. Suppose it is, if you take dry wood, water. Water it is having high water potential. Dry wood having low water potential. So water potential gradient established from are you getting this diagram explanation? Water potential gradient established from water to dry wood. Water, dry wood. So water absorbed by the dry wood, because dry wood it is having low water potential, water it is having high water potential. Okay, the first case. Okay, so in this we do expect there is a difference of water potential between the dry wood and what it is water. Suppose you observe this. Water soaked wood, again placed in water. Water soaked wood, again placed in water. No imbibition. Why no imbibition? Already it absorbed water. There is no difference of water potential between what it is the soaked wood, water soaked wood and water. So no difference of water potential. Are you getting this? The two diagrams. There must be affinity or attraction. Okay. Affinity or attraction. Affinity The two diagrams, you got it? These two. There must be some affinity or attraction between what is imbibed and water to be absorbed. Okay. Okay, it is. Okay. Okay, Venkatesh, Himalakshmi, okay. These diagrams, affinity or it is attraction, okay. Venkatesh, Himalakshmi, rest of the members about the two diagrams, okay. And resins swells in volume. Rubber do not swells in volume. Place it in water. Okay. It is right. Resin swells in volume, placed in water, right? Rubber do not swells in volume, placed in water, right? Because not having imbibing capacity. It is not having imbibing capacity. Okay, it is. And unpainted wooden doors swells in volume in rainy season, Okay, right, it is. Unpainted wooden door swells in volume. It is right. Painted wooden doors is not supposed to absorb water. So unpainted wooden door swells in volume in rainy season. Okay, it is. So resin swells in volume. Imbibition. Rubber do not swells in volume due to lack of or it is imbibition. That is imbibition. And next it is plasmolysis. Before knowing the plasmolysis, suppose if you see, it is the cell, it is external solution, it is cell sap. It is having 0.7 molar concentration. It is having 0.4 molar concentration. So external solution concentration less than that of cell sap. So it is hypotonic solution. What they call it is hypotonic solution. And it is hypertonic solution. The concentration of external solution greater than that of cell sap, what do you call it is hypertonic solution. The cell sap concentration balances with external solution, what they call isotonic solution. Here, cell sap concentration balances with external solution. So external solution said to be what it is isotonic. So isotonic, hypertonic and hypotonic solution. So we can know about the behavior of these cells with regard to the water movement into what it is surrounding medium. That is hypotonic, hypertonic and it is isotonic. Okay. The behavior of this water movement depend upon what external concentration of what is medium. So hypo, hyper and isotonic. Okay, it is. And it is a cell and it is the membrane and it is vacuole, nucleus and it is cytoplasm. If this cell placed in hypertonic solution whose concentration higher than that of cell sap, hypertonic solution. place in hypertonic solution. Corners of the cells, plasma membrane get apart from the corners of the cells. Here, plasma membrane get apart from corners called incipient plasmolysis. What do you call it? Is incipient plasmolysis. And by keeping the cell for some more time, extended in keeping hypertonic solution, extended keeping in hypertonic solution, it is cell wall and it is plasma membrane, it is tonoplast. There is shrinkage of what? Protoplast. There is shrinkage of protoplast. In between the cell wall and membrane, what is present? External solution. The space occupied between cell wall and membrane is external solution. Why? Cell wall is permeable to water and substances dissolved in it. Because cell wall is not going to be a barrier. Okay. So, what is the space occupied by in between the cell wall and membrane? External solution. Why? Cell wall is permeable to water and the substances dissolved in it. Is there any change in case of cell wall? Cell wall, it is rigid. It is not going to change its shape. Okay. As protoplast shrinks, vacuole size decreases, and protoplast size, that is, decreases. So during these changes, water from cytoplasm goes out, then water from vacuole goes out. It is also important. The change, it is water first from cytoplasm goes out, then followed by water from vacuole. The content of the vacuole, it is decreases, that is the size decreases. And size of nucleus is not going to be affected. As cell wall is rigid, it is not going to be any change observed in cell wall. Okay. When a cell placed in hypertonic solution, initially you observe plasma membrane get apart from cell wall at corners. What do you call it? Is incipient plasmolysis. If further extension in hypertonic solution, there is a shrinkage of protoplast because of difference of water potential gradient. In cell it is hypotonic and external solution hypertonic, exosmosis occur. And exosmosis is continuous shrinkage of protoplast occur. So shrinkage of the protoplast when cell placed in hypertonic solution, what do you call it? Is plasmolysis. Okay. Okay. Plasmolysis. And this plasmolyzed cell Sirisha, Sheikh Muhammad Sultan, Afreen, Meghanjali, Venkatesh, Himalakshmi. Okay, it is. And this plasma, what do you call this cell? It is plasmolized cell. Plasmolized cell. This plasmolized cell in hypotonic solution. The plasmolized cell in hypotonic solution. What happens? Cell become turgid. It regain turgidity. Cells regain turgor when placed in hypotonic solution. What do you call it is? Deplasmolysis. So plasmolysis is a reversible phenomena only experimentally but not in nature. In nature it is not a reversible. Experimentally it is reversible. So plasmolysis it is experimental phenomena. Plasmolysis is an experimental phenomena to know about the behavior of the cells with regard to the water movement depend upon what it is concentration of what it is the medium. Okay. It is experimental phenomena and reversible, not a natural phenomena. So, to know about behavior of cells with regard to water movement depends on external medium. It depends upon external medium. External medium it is hypertonic. Cell loses water and protoplast shrinks. So, shrinkage of protoplast when placed in hypertonic solution called plasmolysis. lysis. Which osmosis occur in plasmolysis? X osmosis. X osmosis. In deplasmolysis, end osmosis. Okay. In deplasmolysis, end osmosis. So plasmolysis, X osmosis, deplasmolysis, end osmosis. In plasmolyzed cell, there is no pressure potential. If you take volleyball, remove the air from it, it is not inflated. So there is no pressure potential. If you press from the top, a depression is going to be made. So depression is going to be like this only in volleyball. So like this in this cell, what do you call the condition? It is not a turgid condition. So in plasmolyzed cell, pressure potential is 0. So water potential contribute to what? Solute potential. Water potential is equal to solute potential in plasmolyzed cell. This exosmosis, deplasmolysis, it is endosmosis. And salted pickles. Sugar coated jams. Salted pickles, sugar coated jams last for long time. If pickles are improperly salted, not with the proper concentration, bacterial and fungal spores spoil these pickles. So, salted pickles and sugar coated jams last for long time. The well salted pickles, well sugar coated jam lasts for what it is long time. Suppose if you take A jam, if you take B jam, jams are sugar coated, or this, suppose it is fungal spore, it is bacterial spore. In this, water potential is -5. Water potential is -5. In this, water potential is -15. In this, water potential is -15. Water potential is -5. So there is a difference of water potential. Okay, there is a difference of water potential between spore and jam. So in this, suppose it is X, it is Y. Already mentioned A jam and B jam. In case of A jam, it is having -15, that is MPa, megapascal. That means properly it is sugar coated. And bacterial spores having concentration less than that of what it is A jam. So spore concentration of solute less than a jam. So it undergoes what it is. So undergo X osmosis. So battle sport cannot survive. So A jam going to be fresh. Okay. A jam is going to be fresh. In this concentration, the solute concentration is more in case of the fungal spore. It is not properly sugar coated when compared to the concentration of what the spore. So in this, what type of osmosis? Endosmosis and germination of fungal spore. Bijam is going to be spoiled. Because water always moves from higher water potential to lower water potential. Higher water potential means solute concentration must be low. Okay. I hope you understood with regard to this jam A, jam B. Jam A, jam B. Okay. Jam A, jam B. Okay, it is. Okay, what are the contents? B is going to be spoiled, A is going to be fresh because properly sugar coated, not properly sugar coated as it is supposed to be. Mahesh Reddy, Venkatesh, okay, rest of the members, okay, it is. So, think it is the movement of water from its high concentration to low concentration. So, exosmosis and endosmosis phenomena here. When properly it is salted, bacterial and fungal spores undergo plasmolysis. That means which undergo exosmosis. Instead of it, it is not properly salted, concentration is less, it leads to endosmosis of bacterial and fungal spores. Then it leads to spoilage of pickles. In case of the germs, it is sugar. So well sugar coated jams last for long time than improperly sugar coated jams. There's one point. And properly salted pickles last longer than what it is improperly salted pickles. Okay. Geeta Srinivas, Manasa Murthy, okay. Navya Kinchugunda, okay. Venkatesh, Geeta Srinivas, Himalakshmi, Maheshi Reddy, okay. Shirisha, Manisha, Murali Besta, okay. Venkatesh, Sivaprasad, Pavan Sagar, Chinnu, Venkatesh, okay. Okay, this session over.